Hello everyone, welcome to the ITLS Academy. Today we are going to discuss the topic that is your drug discovery and development. Okay, so before starting the lecture, we have to know that what does this drug means, or you can say that what is the definition of the drug. Okay, we all know that this drugs is defined as any substances except the food, any substances except the food that is used to prevent the diagnose, treat, or relieve the symptom that is used to prevent, diagnose, relieve, or treat these symptoms which are caused by the disease. Just a minute. So symptoms. Symptoms of a symptoms of a disease or any abnormal condition. It simply means that a substance which is used for the treatment of any disease, that is called a drug. Okay. Or in a basic language, we said that it is the medicines, injections. Okay. They are what? They are all come under the category of this drugs. Okay. Drugs also affect the brain and the rest of the body work and cause the changes in the mood, awareness, thoughts, and behavior. Okay, so there are different diseases, and for the treatment of that diseases there are different drugs. But initially these drugs are not discovered. Okay, so um, in today's lecture we are going to discuss about the how these drugs are discovered and how these drugs are developed. Okay, so initially what ayurveda are used for the treatment of the disease. Then this ayurveda also used in the manufacturing of the drug. So in today's lecture we are going to discuss all these things. So let's start today's lecture. Introduction. In the past, most drugs has been discovered either by identifying the active ingredient from the traditional remedies. Okay, so if we talk about the uh, thousands of year back, then there is a traditional remedies are there. Traditional remedies are there for the treatment of the disease. Now, from the past, these drugs are discovered by identifying that the which active ingredients are present at that particular traditional remedies, or by a serendipitous discovery. But now we know disease are controlled at a molecular and physiological level. Okay, but in today's these disease are controlled by a molecular or a physiological level. Also, shape of a molecule at a atomic level is well understood. Okay, information of the human genome. Now come to the next one, history of the drugs. Okay, if we talk about the pre 1919, then at that time herbal drugs are there. Okay, herbal drugs means that ayurveda means that the different trees are used for the treatment of the disease, or you can say that the jari booties, they are used for the treatment of the disease. Next is your serendipitous discovery. Okay, now in 1920s to 1930s, different vitamins are introduced, and these vitamins are used for the deficiency of the deficiencies, or for the treatment of the deficiency caused by that particular vitamins, caused by that particular vitamins. Suppose that someone have a deficiency of the vitamin A. So the doctor are advised to take a vitamin A capsules like that. Okay. And vaccines are also introduced in 1920s and 1930s. Now in 1940s, antibiotics are introduced. Antibiotics are used for the treatment of the disease which are caused by the bacteria. They affect the bacteria. R&D boost due to the WW2. Then in 1950s, new technologies are introduced. DNA are discovered. In 1960s, there is a breakthrough in etiology. In 1970s, there is a rise in the biotechnology and use of the IIT. In 1980s, commercialization of the drugs discovery and combinational chemistry. In 1990s, robotics are introduced. These robots are even there are several operations which are done by a robots. So this is called a robots technology. Automations are also introduced in 1990s. So this is the history of the drug discovery, how these drugs are discovered and in which year these drugs are discovered. Now come to the next slide. Registration. The Ministry of Health and Family Welfare and the Ministry of Chemicals and Fertilizer have a major role in the regulation of the IPM. Okay, so for the regulation of the IPM, there are two ministries are responsible. One is the Ministry of Health and Family Welfare, and another is Ministry of Chemical and Fertilizer. Then NDA must be submit to our DCGI. Phase 3 study reported to a CDL Kolkata. Packed, inserted, approved by a DCI. Marketing are approved by a FDA. Okay. Now I'm going to tell you the term of this. What is the full form of this IPM, DCGI, CDL, DCI, and FDA? Basically, this IPM is what? This IPM is Indian Pharmaceutical Market. Indian Pharmaceutical Market. So it means that the Indian pharmaceutical market are controlled by the Ministry of Health and Family Welfare and the Ministry of Chemical and Fertilizer. Now the next is your DCGI. This DCGI are stand for the this DCGI are stand for the Drugs Controller General of India. Drug Drug Controller General of India. That means that NDA must be submitted to the Drugs Controller General of India. Phase 3 study reported to us CDL, that is your CDA. That the CDL means that Central Drug Laboratory. Central Drug Laboratory. Okay. And FDA, you all know that it is Food and Drug Administration. Food and Drug Administration. Okay, now this NDA means that new drugs application. New drugs application. Okay, if there is any application for a new drug, then it must be submitted to a Drugs Control General of India. Okay, and phase third reported to the Central Drug Laboratory. Okay, and packed are inserted to the DCI. Now DCI means that Drug and Chemical India. Drug and Chemicals India. Okay, so this is how the registration are done. Now the next one, market scenario. Around 800 M spent to bring a $800 So this is talk about in dollar. Around 800 M are spent to bring our new drugs to a market. Okay, if new drugs are spent to the introduced to the market, then around $800 are, and 800 M dollars are required. Around 127 billion dollars are spent in 2010 on a pharmaceutical R&D. Now, CRO share in research generation operation is about 27%, and total world CRO market is around 16.3 billion. Indian share is around $500 million. Okay, this is about the market scenario. Top CROs. The first column is for a contract research organization, and this is your revenue. Quantiles, that is around $2.5 billion. Pharmaceutical product and development, a $1.8 billion. Covance, $1.4 billion. Charles River Laboratories, $1.2 billion. Paraxil, $930 billion. Icon, $887 billion. Kendall, $590 million. Permanent, $470 million. PRA International, $410 million. 4G Pharmacovigilance, $391 million. It is the top CRO's revenue, that is your contract research organization. CRO means that contract research organization. CRO means that contract research organization. Okay, and their revenue. Now the next is Actimus Bioscience, their top CROs in India and their location. So Actimus Bioscience, it is present in Hyderabad. Advinus Therapeutics, Bangalore. Origin Discovery Technologies, Bangalore. Chem Biotech, Kolkata. GVK Bioscience, Hyderabad. Jubilant Organosis, Bangalore. Ranbaxy Life Science, Mumbai. Reliance Life Science, Mumbai. Suvin Life Science, Hyderabad. Syngene, Bangalore. Now the next is most valuable R&D projects. Okay, they are the product and the company and their phase and pharmacological class and today's NPV. The first product is Degludec, company Novo Nordisk, phase 3, pharmacological class insulin 5807. Tofacitinib, company Pfizer, phase 3rd, pharmacological JAK3 inhibitor 4953. BG12, Biogene IDEC, phase 3. Fumarate 4666. Incivec, company name J&J, phase 4, pharmacological class Hep C protease inhibitor 4332. Okay, Rilovir, DR systemine, AMR 101. Eliquis, one is belong from a Pfizer and one is belong from a Bristol Mycel, and both are belong to the phase 4. Bexero Novartis belongs to the phase 4 pharmacological class meningococcal B vaccine and 3250. Now top companies by R&D expenses. The first one, the first column is for the name of the company, and the second column for the R&D is spent uh, in dollar, and this data is from 2010. Novartis 7.9, Merck and Co 8.1, Roche 7.8, GlaxoSmithKline 5.7, Sanofi 5.8, Pfizer 9.1, Johnson and Johnson 4.5, Eli Lilly 4.7, AstraZeneca 4.2, Takeda 3.4, Bayer 2.3, Bristol Myers 3.3, Boehringer Ingelheim 3.1, Amgen 2.8, Novo Nordisk 1.7. Now the next is your drugs development and cost breakup. The first one is your R&D function and their percentage. Discovery basic research, that is first one is your synthetic and extraction. Synthetic and extraction, 10% their contribution. Biological screening and testing 4.2. Sorry, their costing breakup. Biological screening 4.2. The next is your preclinical testing. The first one is your toxicological safety testing 4.5. Pharmaceutical dosage formulation 7.3%. Then clinical trial phase 1, phase 2, phase 3 29.1%, phase 4 11.7%, manufacturing and quality control 8.3%, IND and NDA 4.1%, bioavailability 1.8%, other 9%, and total is 100%. Now the next is your drugs discovery and development timeline. Okay, this the blue phase is your drug discovery, then this green phase is your preclinical, then this purple phase is your clinical trial. Okay, this red phase your FDA review, and this circle is for a FDA approved drug. Okay, so this drug discovery is around 10,000 compound if it's there, then only 250 compounds come under the preclinical trial. Then from 255 compound come under a clinical trial. Then it is approved by the FDA, and one is from FDA approved drug. Okay, so it take from drug discovery and preclinical trial take around 6.5 years. Okay, and this clinical trial take around 7 years. FDA review it take around 1.5 years. So this is the time which are taken from a drug for the discovery. Now the next is your drug discovery. There are different method for the drug discovery. First one is your random screening, then molecular manipulation, molecular designing, drugs metabolite, and serendipity. So these are the five method for the drug discovery. Okay, for now, first of all, you have to select a target, then lead discovery, medicinal chemistry, in vitro studies, in vivo studies, and clinical trial and therapeutic. In target selection, there is a cellular and genetic target, genomic, proteomic, bioinformatics, lead discovery, synthesis and isolation, combina combinatorial chemistry, assay development, and high throughout the screening, medicinal chemistry, library development, SAR studies, in silico screening, and chemical synthesis, in vitro studies, drugs affinited and selectivity, cell disease model, MOA, and lead candidate refinement. In vitro studies, animal model of the disease state, behavior state, functional imaging, and ex vivo state. Then at the last, there is a clinical trial and therapeutic. Now, the next one is your target selection. The first one is your target selection. Target selection in a drug discovery is defined as a decision to focus on finding an agent with a particular biological action that is anticipated to have a therapeutic utility. So first of all, what we have to do, we have to select a target for a drug discovery, and it is defined as a decision which are focused on a finding a particular agent. Either this agent are what they are biological action, and that is antiseptic to have a therapeutical utility, and then it is influenced by a complex balance of a scientific, medical, and strategic consideration. Then after that, after the selection of the target, there is the second point is your target identification. Okay, so this target identification is to identify the molecular target that which are involved in a disease progression. Okay, so target identification means that the identification of the molecular target which are involved in a disease progression. Next is your target validation, which can be defined as that it is used to prove that the manipulating the molecular target which can provide a therapeutic benefit for the patient. Okay, so that means that this target should be beneficial for the patient. Beneficial for the patient. Okay. Now the next is your target selection. Biochemistry classes drugs target G protein couple receptor is around 45%. Enzyme 28%, hormone and factors around 11%, ion channel around 5%, nuclear receptor around 2%, techniques for our target identification. Now the next is your cellular and genetic target. Okay, so in target selection, the first one is your the first technique is your cellular and genetic target, which involve the identification of the function of a potential therapeutic drugs target and their role in a disease process. Okay, so when we decide the target or when we select the target, then the first one is that we have to identify that that the function of that therapeutic drugs target and what is the role of that drugs in a disease process. For a small molecular drug, this step in the process involve identification of the target receptor or a enzyme. If there is a small drug molecule, now in that case, there is a process which involve in a there is a process which involve in a identification of the target receptor and an enzyme. And the next is your some whereas some biological approaches which are focused into a gene or transcription level. Then next is your drugs usually act as a either cellular or a genetic chemical in the body which are known as a target, which are believed to be associate with the drugs, associate with the drugs disease. Now the next is your scientists uses a variety of techniques so that they will isolate the individual target and then study about it. Now, and in the study they study about their function and how they influence the disease. Okay, so there are different compounds which are identified and how they interact with the particular drug target. They will also help in the treatment of the particular type of the disease. Now see what is written in a slide. Scientists uses a variety of technique to identify and isolate the individual target to learn more about their function and how they influence the disease. Compounds are then identified that have a various interaction with the drug's target that might be helpful in the treatment of a specific disease. Now the next one is your genomic. Genomic means that the study of the genes and how what is the function of that particular genes. Okay, the main or aim of the genomic is that what is the structure of the genome and how it is involved in a mapping of the genes and the sequence of the DNA. Okay, see what is written here. The study of the gene and their function. Genomic aim to understand the structure of the genome, including the mapping of the gene and sequencing the DNA. Next is you seek to exploit the finding from the sequencing of the humans and other genomes so that to find a new drugs target. Human genome consists a sequence around 3 billion nucleotides. Okay, if we talk about the human genome, then human genome contain around 3 billion nucleotide. 3 billion nucleotide. That is your ACGT basis, which in turn properly encode around 35,000 to 50,000 of genes. Drews estimate that the number of the genes are implicated in a disease, both those due to the defect in a single genes and those arising from a combination of the genes, about 1,000. So based on 5 to 10 linked protein per gene, he also proposes that the number of the potential drugs targets which lie between 5,000 and 10,000. Okay, so there are single nucleotide polymorphism, that is also called a SNP libraries. They are used to compare that the genome from both healthy and the sick people and to identify where their genome vary. Okay, so in single nucleotide morphism, there is a library in which there is a information of the genome, and the information of the genome of both type of the person, that is your healthy person and the sick person or people. And then they in that library they identify that the how their genome are vary from each other, means that the healthy person genome are how the genome of healthy person are vary from the sick person. Now the next is your proteomics. Proteomics means that the study of the proteome, study of the proteome. It is the complete set of the protein which are produced by a species using the technologies of the large scale protein separation and identification. It is becoming increasingly evident that the complexity of the biological system which are lie at the level of the protein, and that genomic alone will not suffice to understand these system. It is also used that the protein level, that is that disease process become manifest, and at which most of the 90% of the drugs act. Therefore the analysis of this protein, which include the protein nucleic and protein ligand interaction. So there are three type of the interaction. One is your protein interaction, one is your protein nucleic acid interaction, and one is your protein ligand interaction, will be the utmost importance to a target discovery. Proteomics is the systematic high throughout the separation and characterization of the protein within a biological system. Target identification with the proteomics is used to perform by comparing the protein expression level in a normal and a diseased tissue. 2D page is used to separate the proteins which are subsequently identified and fully characterized with the LC-MS and MS. Now next is your bioinformatics. Okay, bioinformatics is a branch of biomolecule that involve a extensive analysis of the biological data. So bioinformatics means that simply means that there is a collection of the there is a collection of biological data in a computer for the purpose of enhancing the biological research, and these biological data are used in a biological research. It also play a various uh, it also play a role in various stages of the drug discovery, like your target identification, computer screening of chemical compound, and pharmacogenomic. Now the next is your this bioinformatics method are also used to transform the new sequence into a meaningful information. Okay, bioinformatics are also used for the transformation of the raw sequence into a meaningful information, that is genes and their encoded protein, and it is used to compare a whole genome, that is a disease or like a person suffer from that particular disease or not. It was also used to compare our entire genome of a pathogenic and non-pathogenic strain of a microbe and identifying the genes protein associated with the pathogenism. Using gene expression micro error array and gene chip technologies, a single device, it can be used to evaluate and compare the expression around 20,000 genes of healthy and diseased individual at once. Lead discovery. After target selection, the first one is your lead discovery, and this lead discovery are divided into four step. The first one is your synthesis and isolation, then combinatorial chemistry, then asset development, and high throughout the screening. The first one is your identification of small molecule modulars of protein function, the process of transformation these into a high lead content lead series. The first one is your synthesis and isolation. Okay, so in synthesis and isolation, the first one is the separation of the mixture, then separation of the impurities, then in vitro chemical synthesis and biosynthetic intermediate. Okay, the next is your combinatorial chemistry. It is a rapid synthesis or a computer simulation of large number of different but structurally related molecules. Okay, so in combinational chemistry, there is a rapid synthesis of a large number of different but a structural related molecule. Search for a new lead, optimization of the target affinity and selectivity, ADME property. It also reduces the toxicity and also eliminate the side effect of that particular compounds. The next is your assay development. This assay development are used for measuring the activity of the drugs. When we have to measure the activity of the drug, we use a assay development. It also discriminate between the compound. It evaluate the express protein target and enzyme substrate interaction. Express the protein target. Now next is your high throughout the screening. Okay, high throughout the screening of drug target against a selection of the chemical and identify of highly target specific compounds. The next is your this is a high throughout the screening diagram. The next is your medicinal chemistry. Medicinal chemistry is defined as a discipline at the intersection of the synthetic organic chemistry and a pharmacology. It focuses on a small organic molecule and not a biological and inorganic compound. It is used in a drug discovery, lead optimization, hit to lead, and process chemistry and development. Library development. In the see this medicinal chemistry are again they have four steps. One is your library development, SAR studies, in silico screening, and chemical synthesis. The first one is your library development. In library development, there is a collection of store chemical along with a associate database, and it also assists a high throughout the screening, and it help in a screening of drug target and based on a organic chemistry. The next is your SAR studies. It help in identification of the pharmacophore. Pharmacophore is the precise section of the molecule that is responsible for a biological activity. Okay, so this pharmacophore are defined as a it is a section which is used for the uh, like identification for a biological activity. Enable the next point is enable. It is enabled to prepare more active compound, and it allow the elimination of a excessive functionality. Now the next is your SAR studies diagram. It is a morphine molecule. The next is your in silico screening. The first point in silico screening is computer is used to stimulate the screening of the chemical. It is help in finding the structure that are most likely to bind to a drug target. Okay, we all know that in a drug there is a drug target drug target space where the molecule are bind. So in silico screening are used to find out that um, that structure in a that structure in a molecule that will bind with the drug target. Filter enormous chemical space, economic than HTS. Next is your chemical synthesis. Chemical synthesis, it involved in the production of the lead compounds, which is suitable for the quantity and qualitative to allow a large scale animal and eventual extensive human clinical trial. Optimization of the chemical route for a bulk industrial production, and it is suitable for the drug formulation. The next is your in vitro studies. In vitro, there again there is a fourth step in vitro studies. The first one is your drug affinity and selectivity, cell disease and models, MOA, lead candidate and refinement. So in glass studies using a component of organism, that is test tube experiment. For example, cells are derived from a multicellular organism. Subcellular component, that is your ribosome and mitochondria. Cellular and subcellular extract, that is your wheat germ reticuli extract, and purify the molecules like DNA and RNA. Okay, advantages of in vitro studies. In vitro studies can completed in shorter period of time, and it also reduces in post clinical trial risk in post clinical trial, and permit an enormous level of the simplification of the system. Investment investigator can focus on a small number of component. Okay, so the first one in vitro studies is drug affinity and selectivity. So drug affinity is defined as the ability of the drug that it will bind with the biological target. This biological target either receptor, enzyme, transporter system, etc. Selectivity are defined as a drug should not bind to a specific receptor site on the cell. That is, for example, aspirin. The next is your cell models. The first one is your isogenic human disease model. They are defined as a family of a cell that are selected or engineered to accurately model the genetics of a specific patient population in vitro. Next is your stem cell disease model, which is defined as a adult or a embryonic stem cell carrying or induced to carry are defective genes which can be used for the investigation in vitro to understand that a latent molecule mechanism and the disease characterization. Next is your lead candidate refinement. It is defined as a optimizing chemical hit for a clinical trial. Okay, so this lead candidate refinement is simply a optimizing chemical which are used for a clinical trial, and it is commonly referred as a lead optimization. Refinement structure is necessary in order to improve the potency, odor availability, selectivity, pharmacokinetics property, and safety ADME property. In the next is your in vivo studies. This in vivo studies are totally experimentation based, and they are using a whole living organism, and it give information about the metabolic profile, toxicology, and drugs interaction. Okay, again it divided into a four step. One is your animal model of disease state, behavior studies, functional imaging, and ex vivo studies. So the first one is your animal model disease state. Test condition involve induced disease or injury which are similar to a human condition. Must be equivalent in a mechanism of a cause. It can predict a human toxicity in 70% of the cases. Example, SCID mice HIV, NOD mice diabetes, Dino-radio D gene function. Behavior studies, there are tools which is used for the investigation of the behavior result of the drugs. It is used to observe a depression and mental disorder. However, self-esteem and a suicidality are hard to induce. For example, despair based force swimming tail suspension, reward based, anxiety based. Next is your functional imaging. There are a method for the detection or measuring the changes in a metabolism, blood flow, regional chemical composition, and absorption. There are traces or probes. Traces are probes or used. Modulators are used. That is your MRI or CT scan. Ex vivo studies, experimentation on a tissue in an artificial environment outside the organism with the minimum alteration of a natural condition, counters ethical issues. Example, measurement of the tissue properties or realistic model for a surgery. Then the last one is your clinical trial. Clinical trial are again, they have four step: phase 1, phase 2, phase 3 and phase 4. There is a set of the procedure which are involved in a medical research and drug development, so that to study the safety and the efficacy of the drug, essential to get a marketing approval from a regulatory authority, and it required around 7 years, that I already told you in the initial slides. Phase 0, recent designation, also known as a human microdosing studies, first in human trial conducted to study a exploratory investigational new drug. It is used to design to speed up the development of a promising drugs, concerned with a preliminary data on our drug pharmacodynamics and pharmacokinetics, efficacy of preclinical studies. Phase 1, clinical pharmacologic evolution, first stage of a testing in a human subject. 20 to 50 volunteers are healthy volunteers are required, and the concern is about the human toxicity, tolerate the dosage ranges, and a pharmacology and dynamics. Type of first trial is SAD and MAD. SAD means that single ascending dose, and MAD is your multiple ascending dose, and the last one is your food effect. Phase 2, control clinical evolution. It required around 50 to 300 patient control single blind techniques, concern about the safety, efficacy, drug toxicity, and drugs interaction. Phase third, Phase third is about the extended clinical trial. They are the most expensive type of the trial, and they are time consuming. Around 250 to 1000 patients are required. Controlled double blind technique, concerned with safety, efficacy, comparison with the other drug, and package insert. Phase 4, most of the marketing surveillance, it is designed to detect any rare or long term adverse effect, adverse drug reaction monitoring, and pharmacovigilance. Okay, this slide already discussed in the starting one. Gene therapy. Gene therapy is the technique for correcting the defect occur in a gene. Suppose that this is your gene. If there is any defect occur in that gene, then there is a technique which is used for the correction of that defect. It is the process of inserting genes into a cell to treat a disease. Gene therapy is used to correct a deficient phenotype. Gene therapy approaches. Germline gene therapy. Okay, somatic gene therapy. In germline gene therapy, if there is any defect occur in a sperm or a egg, then the modified functional genes are introduced and integrate in their genome. Now see what is written here. Sperm or egg are modified by our introduction of a functional gene which are integrated into their genome. Changes would be heritable and would be passed on to a later generation. Okay, if gene therapy are occur in a first generation, then it can also transfer to the later generation because it is heritable. The next is your somatic gene therapy. The therapeutic genes are transferred into the somatic cell of a patient. When functional gene are transferred to the somatic cell of the patient, then that type of the therapy is called a somatic gene therapy. Changes will not be inherited by the patient of a sprint or a later generation. Okay, germline gene therapy are transferred to the later generation, but the somatic gene therapy are not transferred to the later generation. Okay, some type of the this gene therapy. The first one is your vivo gene therapy and in vivo gene therapy. Ex vivo and in vivo. Ex vivo means that the transfer of the therapeutic genes in a culture cell which are then reintroduced into a patient. Okay, so first of all, these therapeutic genes are transferred into a culture genes and then transferred to the patient. This type of the gene therapy is called a ex vivo gene therapy. Now the next is your example is therapy for a ADA deficiency. Next is your in vivo gene therapy, in that type of the gene therapy, that the genes are directly introduced into a cell of the particular tissue, which is referred to as a vivo gene therapy. For example, therapy for a cystic fibrosis. Gene therapy vectors, viruses, that is your retroviruses, adenoviruses, adeno-associated viruses, herpes simplex viruses, pure DNA construct, lipoplexes, DNA molecule conjugated, human artificial chromosome. There is a limitation for a gene therapy. They are short-lived nature of the gene therapy, immune response, problem with the viral vector, and the multigene disorders. Some recent development, that is your nanotechnology plus gene therapy yield. It is used for the treatment of the torpedo cancer. Result of world first gene therapy for inherited blindness show a sight improvement. There is a new method of a gene therapy are alter immune cell for treatment of the advanced melanoma. Dual gene therapy are used in a suppressed lung cancer in a preclinical test. Orphan drug is a pharmaceutical agent that is used, that has been developed especially to treat a rare medication condition, that condition itself being referred to as a orphan disease. Okay, so orphan drugs are simply used for the treatment of the orphan disease. This is a national organization for a rare disorder, not European Organization for a Rare Disease, Euro RDIS. Advantages: tax incentive, enhanced patient protection and marketing rights, clinical research and financial subsidization, rise in research and development, Crown Corporation. Orphan Drugs Act was passed in 4th January 1983. In that time there are 6,000 orphan there are 6,000 person who are suffer from the orphan diseases. Unprofitable drugs development, they are affecting more than 2 lakh person. Orphan drugs states to 1090 drugs, and there is an amendment in 1985, marketing and exclusivity. FDA orphan drugs approved. Okay, so this is a big pharma, then a small pharma, established biopharma, and small and medium pharma. Now the next is your rare disease and orphan drugs. So some rare disease which are occur in a person and their causes and their orphan drugs. That is your Gaucher disease, and orphan drugs is Miglus state, Fabry's disease, and so on. And the last one is your SCID, that is cured by a orphan drugs called a pegadricase. So this is all about the drugs development and discovery of the drug. I hope you will understand, but if you have any query related to this topic, you can ask me in a comment box, or for a further detail you can follow the ITLS Academy on different platform like Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and subscribe our channel ITLS Academy on YouTube, and get our app from a Google Play. Or if you have any query, you can ask your query to the helpline number that are already mentioned in your slide, that is 7080833450. Thank you for watching. Please like, share, and subscribe our channel, and visit our website, that is www.itlsacademy.com.